Welcome to the Explainer. Today, we're going to break down a big one, a 12-mark question all about monoclonal antibodies from an AQA GCSE Biology paper. We're going to pull it apart piece by piece so you know exactly how to nail every single mark.
So here's the plan. We'll look at the whole question, then we'll break down each part and see exactly what the mark scheme is looking for. And to finish, we'll pull out the key takeaways that'll help you crush these long questions in your own exam.
Okay, so first things first, let's get our heads around the main topic. This entire question revolves around monoclonal antibodies, or MBABs for short. Just think of them as these super specific, lab-made antibodies that are designed to target one single thing, like a virus, or in this case, a fungus.
So here's the scenario they've given us. Scientists have created new monoclonal antibodies using human cells to fight this fungal infection, Candida albicans. Now pay really close attention to that detail. Human cells, not the usual mouse cells. Trust me, that's going to be super important later on.
Right, let's jump into the first part of the question. This is what we call procedural task, and it's worth a solid 3 marks. The command word here is describe, and that is a massive clue. It basically means tell me the steps. We don't need to get into the why it works, just the how. How do you actually do it to make this invisible pathogen light up and become visible?
For the first mark, you have to say that you bind the fluorescent dye to the antibodies. You can't just mix them together. For your second mark, you then add these new glowing antibodies to the slide, and this part's important. You rinse off any extras that don't bind. This just clears away the noise. And for the third and final mark, the antibodies that have latched onto the pathogen will now glow under the microscope. 3 simple steps, 3 easy marks.
Okay, next up, the question is shifting gears now. We're moving from just finding the pathogen with MBAs to actually treating an infection inside the body. It's asking us to explain what happens.
Did you notice that? The command word changed. It's now explain. That's your signal to connect the dots. You need to link a cause to an effect. It's not enough to just say what's happening. You have to explain why it helps the patient to get both marks. So, to get both marks, you need a 2-part answer. First, the direct cause. More of the pathogens are getting engulfed and destroyed by phagocytes. That gets you 1 mark. Then you have to explain the effect of that. Therefore, there's less damage to the patient's cells, tissues, or organs. Cause and effect, 1 mark for each part.
And now for the main event, the 6-mark question. These ones are the big point scorers, but they demand a really structured, detailed, and logical answer. And this one is all about clinical trials.
We're back to that command word describe, so we need to lay out the whole process of a clinical trial in a logical sequence. You have to think of it like telling a story. You can't skip to the end. You've got to start with safety before you can even think about finding out if it works. Listen, here's the secret to a top-level answer on these big questions. You have to think in clear stages. What absolutely has to happen first? That logical flow is everything. You have got to show that you understand you establish safety before you test for effectiveness. Now this is really interesting. Look at how the mark scheme structures the points. Stage 1. It's all about safety. You give a very low dose to healthy volunteers just to check for side effects. Stage 2, then you move on to patients who actually have the disease to find the best dose and to see if it even works. And stage 3, this is for the top marks. You describe the method. You have to mention using a placebo in a double-blind trial where nobody, not even the doctors, knows who's getting the real drug. Nailing these stages in order is how you get all 6 marks.
All right, now for the final part. It's only worth 1 mark, but it's such a clever little question because it loops all the way back to the information they gave us at the very start.
The command word here is suggest. That means you need to take your biology knowledge and apply it to this specific situation. So think about it. Why would antibodies made from human cells be better for treating humans than antibodies made from mouse cells? And this slide just illustrates the point perfectly. There are actually a few right answers here. The main idea, the core concept, is that the human immune system is way less likely to see human antibodies as foreign and try to reject them. You could also get the mark by saying that because the original lymphocytes came from a human who already beat the infection, we know they're effective. Any of those answers will do the trick.
So after breaking all that down, what are the big lessons here? What's the key to acing a really complex multi-part question like this one?
Honestly, the most crucial thing is to pay super close attention to the command words. Describe, explain, and suggest are not the same thing.
Describe wants a step-by-step story. Explain wants a because. It wants the reason why. And suggest wants you to apply what you know to a new scenario. Understanding these differences is absolutely the key to giving the examiner exactly what they're looking for.
So you see, it's not just about knowing the biology, it's about knowing the rules of the game. Once you know how to deconstruct a question like this, you can use that same logic on almost any other question they throw at you. The only real question is, which one are you going to tackle next?